Hello, everybody, and welcome back to Creation Myths. I got a big one for you today that I'm really excited to go through. Today, we are talking about the creation myth that Winston Ewert's dependency graph model does a better job explaining the existing pattern of biodiversity than common ancestry. And that's a mouthful, so we're just going to shorten that up to creation myth, dependency graphs. And before we go any further, you'll notice that we're doing this in the old format, with me really small in the corner, and we're doing it this way because there's a lot to talk about today, so I want to make sure we have all this real estate where I can show you stuff. So let's start with Winston Ewert himself. Dr. Winston Ewert is a software engineer, so for those of you whose Salem hypothesis alarms just went off, there you go. He has a BS in computer science from Trinity Western University, an MS in computer science from Baylor University, and a PhD in electrical and computer engineering, also from Baylor. He is a senior fellow at Discovery Institute Center for Science and Culture, and he was a senior research scientist at Biologic Institute, but Biologic seems to have shut down in 2021, as far as anyone can tell, so I don't know if that's still considered an active position. Now, before we get into the actual model and what the comparisons are, let's just introduce the concept of dependency graphs, because this was something that was new for me when I first read the articles we're going to talk about, however many years ago it was that I first read these articles. So a dependency graph is a visual representation of dependency relationships, thing one requires thing two, among objects or components in a system. A lot of times you're going to use this in software engineering, where you have a particular module that does a thing, and then you have other components of your software that are dependent upon that module. So here is a simple dependency graph with 4 modules, and in a dependency graph, the arrows indicate dependency relationships. So in this dependency graph right here, D is dependent upon both B and C is dependent upon A and B, and then B is dependent upon A, right? So that is a simple dependency graph, and as we'll see, we're going to apply this type of thinking to biological systems, where instead of a relationship that develops due to common ancestry, you have patterns of similarity that are due to dependence on specific designed modules that are then employed to in the development, I guess you would say, of different individual organisms. Okay, so the argument, the central claim that we hear from Dr. Winston Ewert in the articles that we're going to talk about is that dependency graphs do a better job explaining the pattern of biological similarity that we see in the world today than common ancestry does, as explained via phylogenetics, right? A branching pattern of common ancestors that exist in the more distant past as you work backwards on your phylogenetic tree. Okay, so in other words, this type of representation, which is a dependency graph from the 2018 article we're going to talk about in a few minutes, this type of representation does a better job explaining the biodiversity of these groups than this, a phylogeny. So in a phylogenetic tree, you have your common ancestors, which are represented by nodes, and then you have your descendants from those common ancestors. So here we have nodes representing common ancestors, or MRCA is most recent common ancestor, and we have all the descendant species here. And then in the dependency graph model, we're adding modules for, in this example, echolocation and being marine, living in the water, and this supposedly does a better job explaining the pattern of similarity here compared to this. Right? That's the central argument that we're going to be evaluating. Okay, so before we go any further, let's just talk for a few minutes about phylogenetics, how it works, what we do, how to read a phylogenetic tree, and then we can get into comparing that to the dependency graph model and see how they stack up against each other. So a phylogenetic tree, which you can see a simple one right here, is a visual representation of evolutionary histories and relatedness among groups of organisms. The whole point is you're looking at a branching pattern from common ancestors to your extant taxa, your living groups, the groups that exist today, right? And the point is that the pattern of similarities and the pattern of differences is due to their divergence from their shared common ancestors. So if you look at this simple phylogenetic tree right here, we have 5 species, or 5 taxa, you might say, A, B, C, D, E. At one end of the tree, we have the root. That indicates the most recent common ancestor of all of the taxa on the tree. So that's like the most distant common ancestor on the tree. And then the branch points, which are also called nodes, represent speciation events or divergence events. Each line represents a population or lineage, and then when you hit a branch point, that lineage diverges into 2 separate groups, and you have these series of divergence or series of speciation events that leads to the existing biodiversity. Now, another way to think of that is you could work backwards from the present instead of forwards from the past. And when we conceptualize it like that, we could think about those nodes a little differently. Instead of representing branch points, right, speciation events, splitting events, if you're working backwards from the present, those nodes represent the convergence points. The nodes represent the most recent common ancestor, or MRCA, of the descendant groups. So this node right here is the most recent common ancestor of A and B. This one right here is the most recent common ancestor for C, D, and E, and so on. Okay, so that's basically how you read a phylogenetic tree. Now, how do we make this? Well, what we do now, because we have DNA sequencing and computers and stuff, we're going to take DNA or RNA sequences, depending on the organism you're looking at. My background is viruses, so there's a lot of RNA viruses out there, so you do RNA alignments rather than DNA. But you're going to sequence the genomes or part of the genomes. You're going to align those sequences, and you can see a simple alignment right here. You use various programs to align the nucleotides, and they're going to be color-coded like this, so it's really easy to see how they line up with each other. And then you're going to compare, and you do this computationally, you don't do this by hand, a computer does this. You're going to compare ancestral and derived traits among those sequences in your alignment. Now, an ancestral trait is a trait that is present outside of a group of interest. And when you're looking at like a DNA sequence, an ancestral trait is going to just be a nucleotide, right? A base, A, T, C, or G, that exists outside of whatever specific taxa it is that you're looking at, right? So you're looking at, let's say, for example, mammals, and at a place in the mammal genome, there's T, a thymine. Well, if you were to look at the reptiles and they have that same gene and there's a thymine at that particular site, then that's an ancestral trait, right? It exists outside of the group that you care about. Now, the opposite of that is a derived trait. That's something new. That's a trait, like, for example, a specific mutation that appears in the most recent common ancestor of your group of interest. So sticking with the mammals versus reptiles example, if you have another site where it's an A in the mammals, but it's a G in the reptiles, and you go, I wonder which one is ancestral, and you go to the amphibians and they have the same gene and it's a G there too. Now you can say, oh, the mammals with their A, that's the derived trait. That's the new mutation, and the other groups have the old trait, in this case, the old nucleotide base at that site, right? So those are ancestral and derived traits, and we want to be able to determine whether a particular trait, which in this case means just a nucleotide base, whether a particular trait is ancestral and derived, because that tells us the direction of the evolutionary change, right? That tells us that something new happened as opposed to something old stayed the same. So once we do our alignment, we use a computer, and there's lots of different ways to do this. We're not going to get into the different methods, but mostly we're going to be using some kind of Bayesian approach here, where you're going to take your sequence alignment, you're going to evaluate the differences and score those differences according to some parameters like the population growth model or the nucleotide substitution model, the different relative rates of mutations that happen to different nucleotides. And then you're going to build your phylogenetic tree based on parameters like which is the most likely, or which requires the fewest evolutionary changes. That's called maximum parsimony, right? So you're going to use a computer to score literally thousands and thousands and thousands of different trees according to these parameters and construct the best one based on your data. So that's a very quick, highly simplified overview of how you're approaching phylogenetics to take your sequence data and generate your hypothesized relationship among those taxa. That's the big idea there. Creationists, of course, have lots of objections to the entire practice of phylogenetics. They'll say things like, those are just lines on a page. That's full of evolutionary assumptions. That stuff is wrong, and I want to get that out of the way just right off the bat, okay? So first, phylogenetics, these techniques have been experimentally validated. For example, you can look at a paper by Hillis et al. 1991. I'll link this down below. This is where they used bacteriophages, and they split the populations in the lab. So they had a population, and they split it, and they split it, and they split it, and they generated a bunch of different descendant populations. And then they sequenced those, and they built the phylogenetic tree from the sequence data, and then compared the trees that they built to the actual phylogenetic history of those experimental populations. And all the techniques worked really well. So you can see here, here's the actual phylogenetic tree. Here's the one that they generated based on the phylogenetic techniques. You can see that the relationships are the same. Here is a graph showing how the different types of techniques scored, and you can see they all did really well. This is Clade's resolve, so basically what percentage of groups did they get correct, and it's all in the 80 to 90% range except for this last one. Now, I'll say here that restriction sites are a terrible way to do this. We don't do this anymore because this is from the early 90s. It's all with the sequence data now. And these techniques have only gotten better and more precise, obviously, since the early 90s. So these techniques are perfectly valid. They work. They've been experimentally validated. In addition to that, if you have a problem with your data, if you are trying to use data that are inappropriate, something is going to go wrong. And to show you an example of this, I get to use a paper that was actually written by some of my colleagues, one of my colleagues and my advisor when I was in graduate school. And the point I want to illustrate here is that phylogenetics is not based on just guessing how the evolution would have happened. Phylogenetics is based on directly observed parameters, things like the dates of the isolation of the samples you're using, like when did you collect that sequence, and what are the substitution rates, how quickly do the different mutations accumulate in that sequence. If something is wrong, your output doesn't make any sense. And this figure on screen right now is a great example of this. I love this paper. I was in grad school, and this is Hicks and Duffy were two of my colleagues. Dr. Duffy was my PhD advisor. And so they were working on this virus called RHDV, rabbit hemorrhagic disease virus, and getting really weird numbers. And you can see this in red down here, really weird numbers for the nucleotide substitution rate, and really unusually large error bars for the year of emergence for this virus. Like, when did this virus first appear? We're getting error bars that were spanning 3 centuries, and that doesn't make any sense. And what they were able to figure out is one of the samples, just literally one sample of all of the samples they used, was dated incorrectly. So you had a certain number of substitutions and a certain number of years, and those two numbers were not correct relative to each other because the date was wrong. And when that was the case, when you tried to do these techniques with the sample that was misdated, it totally threw off the entire analysis. That's what you see in red with these error bars that are way too big and with the substitution rate that is way too low. Now, They went and figured out which sequence it was that was incorrectly dated, removed it, and then you get the blue data where the substitution rate is what you would expect it to be, and the year of emergence we could very confidently pin to like the mid 20th century with very small error bars, right? So this is just to show you that these techniques are not arbitrary. They're not based on just, oh, let me just do it in a way that's convenient. They're actually based on concrete observed parameters. And if something is wrong about those, the analysis doesn't work. So I want to get that across to everybody before we go any further, that phylogenetic techniques are to some extent self-checking and self-correcting. Because if there's a problem with what you're doing, you get gibberish. You get an output that makes no sense. So now that we've established our background for phylogenetics, let's look at Dr. Winston Ewert's argument for his alternative, right? Basically, the way he makes his case is to say that some data do not follow the nested hierarchical pattern that you see among living things. There is, as he puts it, an approximate nested hierarchical pattern, but there are also violations of that pattern. So overall, biodiversity is better explained as a series of dependency relationships rather than descent from common ancestors. So again, here is the dependency graph that he uses as an example in the 2018 article that we'll talk about shortly, where he's saying the fact that echolocation appears several times, mammals become marine mammals several times. You've got like the dolphin, seal, and manatee are all in separate clades, but they all became marine. So rather than say that set of traits evolved 3 times, we could say the marine module was employed in each of those cases, right? This is, in effect, a formalized version of the common design argument, right? That we see reused components because a designer reused them as the designer was designing the different groups of organisms. And this is not me putting words in Winston Ewert's mouth. This is how he describes it, okay? So basically, he is claiming to provide a mechanism for common design. So he actually writes in the paper that the problem with common design is the lack of some concrete mechanism. He writes, common design leads to similarity because designers frequently reuse components, modules, and solutions, leading to similarity. However, no hypothesis has been put forth to explain why this non-random reusage of parts would exhibit the appearance of a nested hierarchy pattern. He then goes on to explain how the dependency graph model provides the answer to that question. And I know this is a lot, but I'm going to read this highlighted part because this is important here. So he says, we propose a new hypothesis to explain the approximate nested hierarchy pattern, the dependency graph. This hypothesis is drawn from the techniques used to reuse code in software development. Briefly, instead of a species descending from a single ancestral species, at least in the conventional account, each depends on multiple modules which themselves may depend on other modules. This brings together many of the ideas proposed by design-oriented critics of common descent. It uses the idea of common design by having modules reused in different species. It also draws on functional constraints by having the valid combinations of modules restricted by which modules depend on other modules. Furthermore, it predicts an extended pattern which is the result of the proposed dependency graph mechanism. So to be clear here, what he's saying is we have an approximate nested hierarchical pattern, but there are aspects of living things that don't follow that pattern. So instead of explaining what we see via common ancestry, we're going to explain it via common design, where instead of saying the mammal common ancestor, we're saying the mammal module was employed in the different separately designed mammal lineages. So that mechanism of design explains the hierarchical pattern, but you can also have modules like echolocation or marine mammals that are employed in separate lineages that explain the non-hierarchical parts of the pattern as well, right? So that's what Ewert is proposing to add to this conversation here. So let's just be clear before we go any further that common design doesn't work. I just want to make sure that's out there. The extant pattern of similarity that we see precludes common design. Okay, the pattern of similarity in genome sequences is impossible to achieve via common design, right? The observed nested hierarchical pattern, particularly in the unconstrained regions, is impossible via common design and subsequent evolution, because no matter how you design it, the subsequent evolution is going to disrupt that pattern. The only way to rescue that is by active intervention by the designer, and that's unfalsifiable. Now, I've talked about this at length. There's going to be some resources on this link down below, but I just want to make sure we're clear before we go any further that common design doesn't work. You can't explain the existing pattern via common design unless you are proposing ongoing divine intervention to maintain the pattern from the creation event to the present in every separately created lineage. Just want to make sure that's clear before we move on. All right, so now let's talk about the papers that Winston Ewert wrote on this topic. He has published 2 articles on this topic, and I have them in quotes, the papers, because they're both in Biocomplexity, the Discovery Institute in-house journal. This is not a real journal. It's not real peer review. We've talked about this before. I'm not going to spend time on it, but just go through real peer review, publish in a real journal. Other creationists have done this. I don't know if Dr. Ewert considers himself a creationist. I don't really care. It's either way. Intelligent design is a flavor of creationism. It doesn't matter. Other professional ID creationists, whatever you want to call yourselves, have published in real journals. So Basner and Sanford, 2018, Axe, 2005, Behe and Snoke, 2005. There have been several. I know Beckley and Hosger have some stuff. There have been a bunch. If you want to put a new idea out there into the world, do it in a real journal with real peer review where people are not have a prior ideological inclination to the idea you are proposing. The point of review is that people are going to check your work and challenge it, not be inclined to move it along, right? As a starting position. So I would start there. I would like to see this attempt to go through real peer review. So we're going to talk about these articles, but keep in mind, these are not the same thing as, for example, Basner and Sanford 2018, which for all its problems, was a real peer-reviewed article in a real peer-reviewed journal, which is not the case here. Moving on. Here are the 2 papers. We have in 2018, the dependency graph of life, and then in 2023, aminograph analysis of the auditory protein Preston from bats and whales reveals a dependency graph signal that is missed by the standard convergence model. Now remember, the claim that we're going to be looking at here is that dependency graphs do a better job explaining phylogenetic patterns than common ancestry. Now let's talk about why it's wrong. Let's talk about why this is completely wrong. So now for the fun part. We're going to do this in 2 sections. We're going to break it up, the 2018 paper, the 2023 paper. We'll spend most of our time on the first one because there's just more to talk about there. The dealing with the 2023 one is relatively quick. So the 2018 paper is called The Dependency Graph of Life. That's a play on the phrase the tree of life. And in this paper, we're claiming to test the dependency graph model against the common ancestry model. Ewert uses a Bayesian approach, which is good, like Bayesian statistics here, to compare the probabilities of the pattern of shared gene families via common ancestry and dependency graphs. And surprise, he found that the dependency graph model better fits the data than the common ancestry model. Okay, so before we go any further, let's briefly talk about gene families. A gene family is a group of genes that share common ancestry. So a really good example of this shown on screen is the beta globin gene family in birds and mammals. This is a component of hemoglobin. And we can look at 2 different types of genes that are homologous within a gene family. So you have paralogs that exist due to gene duplication. So for example, an ancestral globin gene duplicated, and that's how we get our alpha globin and our beta globin genes, right? So it's one gene and then it duplicates and now you have two. Those are called paralogs. And then when you have speciation events, that results in orthologs. So the beta globin gene in Homo sapiens, the beta globin gene in chickens, in chimpanzees, right? Whatever. When you have a gene that's in a common ancestor and then that population splits, and now that gene exists in both descendant lineages, those genes are orthologs. They're orthologous genes. It's a type of homologous gene. So that's what a gene family is. And so Ewert is looking at the pattern of the presence or absence of gene families in different groups and evaluating the probability based on the two different models. So that's what's going on here. Okay, so there are three major problems with this paper, and we're going to go through them from smallest to largest. The first is that violations of a perfect nested hierarchy are not evidence against common ancestry. Ewert treats any discrepancies as a problem for common ancestry. So I'm going to read this part. It's a little bit long. I'm going to read it. He says, even amongst more complex life forms, data exist which are not congruent with the hierarchical pattern. Some push for embracing a view of evolution no longer constrained by the tree of life. Those who do not consider the tree of life falsified nevertheless hold to a modified version of it. Mechanisms have been added to explain deviations from the hierarchy, such as horizontal gene transfer, incomplete lineage sorting, differential gene loss, gene resurrection, gene flow, and convergent evolution. He then goes on to say a little bit later, defenders of evolutionary theory still commonly invoke the nested hierarchy as a successful prediction of and compelling evidence for common descent. Right? So the idea here is that we should be looking at the tree of life as envisioned as a nested hierarchical pattern of similarity as basically falsified, even though there are some dead enders who still cling to it. And let's just be clear right off the bat, that's wrong. Most data does actually conform to a nested hierarchical pattern. So he's kind of overselling the case here. There are exceptions to it, but it's not as much as he's presenting it to be. More importantly than that, evolutionary theory explains the discrepancies. It explains the data that violate the nested hierarchical pattern. There are many known mechanisms for this, and we can directly observe and document, and in some cases even predict the occurrence of those events. So one such mechanism is convergence. We see convergent evolution all the time, and it in no way invalidates common ancestry. So for example, here's a figure from a 2002 paper showing multiple emergences of MRSA. Okay, this is methicillin-resistant Staphylococcus aureus. So here is the phylogenetic tree of this bacteria, and each of the lineages with a black dot is a MRSA lineage, so methicillin resistance. Obviously, that trait has evolved multiple times independently. Does anyone think this means that Staphylococcus aureus doesn't have a single common ancestor? No, this is silly. It's silly to point to convergence and say that for some reason that makes one question the common ancestry of the lineages involved, right? That doesn't make any sense. We can also look at horizontal gene transfer, which is the movement of genetic material between individuals. Now, to be clear, before I go any further, just want to be clear that Ewert says we're only looking at animals. Horizontal gene transfer is fairly rare in animals as far as we can tell, so we really don't have to worry about it. And fair enough, it is less common in animals than in other groups, but it's not unheard of, and especially it's not unheard of in the animals that do things like eat algae and practice kleptoplasty, for example, which is where you steal the plastids from your photosynthetic food and use those for a little bit. We've actually documented horizontal gene transfer from the green algae to animals in at least one case. So it's not something that you can completely write off, especially when you're looking at large clades like phyla or kingdom level, as opposed to individual species, which is what Ewert looks at in this paper. So here is how horizontal gene transfer affects the phylogenetic signal. Okay, so here we have two scenarios where on the left for each one, we have the actual speciation tree where lineage 3 branches off first, and then 1 and 2 split from each other more recently. And then we have gene A and gene B where they follow the same pattern as the populations as a whole. Cool. In scenario 2, we have horizontal gene transfer of gene B from lineage 2 to lineage 3, which means this node right here, the branch point of 1 and 2, becomes the most recent common ancestor for gene B. So we get the expected phylogeny for gene A, but we get a different phylogeny for gene B due to the horizontal gene transfer between 2 and 3. So the transferred region is obviously notably different in terms of its phylogenetic signal compared to the rest of the genome that didn't undergo the horizontal gene transfer. We can identify this. We know that regions or genes have unique evolutionary histories. This does not make common ancestry less probable. You observe something like this and say, oh, that's horizontal gene transfer. That's not like a thing that invalidates common ancestry of the lineages involved. Right? And then finally, the last one we'll look at briefly here is incomplete lineage sorting. This is when the phylogenetic history of a gene or allele differs from the relatedness of the species that possess that gene or allele. And this occurs because lineages can branch in a pattern that is different from the pattern of divergence of copies of a gene. Okay, so here we have two scenarios, A and B, where in scenario A, the gene showed with the colored lines here follows the same pattern as the population, which is the gray kind of tubes that contain the gene, right? The gray background. So here you can see it's blue, that's number 3, and then 1 and 2 over here, where the gene, the blue gene or blue allele splits off first, and then the green and the pink split off subsequently. But you could also have a situation like this, where the population follows the same pattern, right? It's 3 and then 1 and 2. But if you look at the pattern of the genes diverging from each other, the pink one branches first, then the green and the blue split. So you get a different phylogenetic tree based on that gene compared to what we saw in panel A right here. That's called incomplete lineage sorting. The neat thing is you could actually predict this based on the divergence times of related lineages. And when you have several divergence events in a short time, you expect and can predict a higher level of incomplete lineage sorting. You could actually predict the exact amount of incomplete lineage sorting you expect to see. And the human-chimp-gorilla divergence is a really good example of this, where somewhere in the neighborhood of 25% of the genome exhibits this pattern, where, for example, chimps and gorillas are more similar to each other than either is to humans. And that's because the sequence of divergence, where it was gorilla and then human and chimp from each other, that was relatively quick. So you have a bunch of divergence events and then 3 lineages after that. That leads to a prediction of the pattern that we see in terms of incomplete lineage sorting. So far from being something that is a problem for common ancestry, we could actually predict incomplete lineage sorting and then check to see if it matches up with our understanding of the shared evolutionary relationships. And the human-chimp-gorilla case is a really excellent example of that. So the point is that none of these mechanisms that disrupt the expected hierarchical pattern are actually a problem for common ancestry, despite what Ewert claims or how Ewert treats them in this paper. But that is just the first of the 3 problems here. The second one, which is a bigger problem, is incomplete genome annotation. Because remember, Winston Ewert is comparing the presence or absence of gene families in the species he's looking at. If a gene family is present in taxa where it is not actually reported, like we have the sequence, but we haven't annotated it, so we haven't said this gene is here, that gene is there for the entire genome, then the analysis is invalid because the pattern that he is evaluating doesn't reflect the biological reality. And to illustrate this, we can look at a simple phylogeny right here where we have in red members of a gene family. And according to the dependency graph model, because this gene family is kind of spread out across different lineages with a relatively distant common ancestor, you would say the dependency model is a better fit to these data, right? You could say there's a module for that gene family that was employed in this lineage right here, and in this lineage that then branched into these two, or you could say separately in each of them. Either way, I'm doing the more parsimonious thing and saying it would be in the common ancestor, but it doesn't matter for the point we're making here, right? So this is how you would employ the dependency graph model to explain these data. But what if these data are incomplete? What if this tree is missing the presence of members of that family? So for example, you could be missing precursor sequences. The precursor sequences would be missing the derived function, right? If you're defining this based on having a gene that does a function, well, a non-protein coding region won't have that function, but the region itself might still be present. This is a really easy error to make when you're lacking a telltale sequence motif, like a very specific little sequence region where it's absent in the non-functional precursor or the precursor that does a different job. If that precursor is non-protein coding, the thing that you're missing could be something as simple as a start codon and a stop codon. And if you don't have those, you miss it in your annotation. But the sequence itself is still there in its ancestral form. The other thing that can happen is you can miss the derived non-functional pseudogenes. And that's what we're showing in pink right here. This lineage and this lineage experienced mutations that inactivated this particular gene. So if you're annotating those genomes based on the presence or absence of the functional gene, well, the pseudogenes aren't going to show up in that annotation, but they're still there, right? The sequences are still present. So now, if you look at this, the dependency graph fails, right, compared to the common ancestry. The sequences have a nested hierarchy, right? You get this neat nested hierarchical pattern exactly as predicted via common ancestry, even if the functional genes are absent. So now, having that dependency module present doesn't actually do a better job explaining that pattern. It does a worse job. Because here's the pattern that we can actually see, where we have the precursor sequence in that most recent common ancestor. It doesn't acquire the function in one lineage, it does in the opposite lineage, and then subsequently, members of that lineage lose the function and it becomes a pseudogene. And we can document each step of that process by looking at the specific sequences that are present rather than just what's in red, the presence or absence of the gene family. All right, so by looking at gene families, incomplete genome annotation is going to miss data, and those data, the actual sequences that are present, are going to show you a different picture. They're going to favor common ancestry over the dependency graph model. And that brings me, speaking of sequences, to the third and largest problem with this analysis. Winston Ewert is making an apples and oranges comparison. Phylogenetic trees and the resulting nested hierarchies are based on actual genome sequences, not the presence or absence of gene families. Ewert uses the latter and says the trees made by the former don't match as well. Well, duh, he's not comparing the same data sets. We don't make phylogenetic trees based on gene families. We make them based on sequence alignments and comparisons. He's taking a different data set and saying the answer isn't as good when we use the mainstream techniques. Well, yeah, you're not using the same data. He's taking trees based on sequence alignments, using the presence or absence of gene families to do a different analysis, and saying that analysis calls into question the first one. How does that even make sense? This is the big reason the dependency model doesn't live up to the claims. It's never been tested against the actual phylogenetic data Ewert claims it does a better job explaining. And to his credit, Dr. Ewert is clear about this. He says, I haven't tested this against sequence alignments. That's a shortfall. That needs to be done in the future. Okay, great. But then it is inappropriate to claim that your model is better than the common ancestry model when you're not even using the same data set. Right? That is not a reasonable conclusion to draw. And bringing this back to something I said earlier, had this gone through actual peer review, that would have been a point that was raised. And the way the conclusions were stated would have been tuned down a little bit, right? Wouldn't have been so hyperbolic. Or the reviewers would have demanded that he do the actual analysis, right? That's the other thing that could have been done there. But we're not done. There are additional problems with this paper. For example, the modules are assigned arbitrarily. You could use a module to explain any non-hierarchical data. And then almost by definition, the dependency graph model is going to be superior to the common ancestry model just in terms of what percentage of the data is it able to explain. The example that Dr. Ewert uses is looking at, I think there were 19 gene families, I think was the number, that were found only in zebra finches and zebra something else with the word zebra in it. That was zebra fish, I think it was. It was zebra finches and zebra fish. I forget. And saying, well, sure, you could just explain that with a module. Okay, but now it's just a totally ad hoc model where you're just throwing modules around to explain any pattern that you claim doesn't have a better explanation. That doesn't actually help us. And again, to Dr. Ewert's credit, he does say this does increase the complexity of the model. That needs to be evaluated, but I don't think he grapples with that fully. I think there is not full accounting of the level of arbitrariness involved in proposing the modules to explain non-hierarchical data. The second problem here is that there is no consideration of the evolutionary mechanisms I described earlier that can lead to a non-hierarchical pattern. Right? So he wrote, and again, sorry for the long quote, but I want to make sure I'm getting the words right. He wrote, "Note that we do not claim that the dependency graph is the only way to produce data which fits the dependency graph better than a tree. Given the wide variety of mechanisms that might be invoked along with common descent, it would not be surprising that some combination of those could produce data which fit a dependency graph better than a tree. In such a case, we would need to separately compare how that alternative evolutionary hypothesis performs against the dependency graph." So what he calls the alternative evolutionary hypothesis is just the evolutionary hypothesis, because we include all those other mechanisms when we do this stuff. So he's not actually testing dependency graphs against common descent. He's evaluating a subset of evolutionary processes, just vertical transmission of inherited traits. This is similar to like what Michael Behe does with irreducible complexity, where he writes, well, if you have a trait that requires multiple mutations, and you have to have each mutation occur in a row, and each mutation confers a small fitness gain, that's not how evolution works, right? So in a similar way to how Michael Behe is omitting many evolutionary processes in describing the difficulty of evolving irreducibly complex traits, which have been directly observed to evolve and is not actually a problem for evolution. In that same way, Winston Ewert here is saying, well, if you restrict evolutionary processes to vertical transmission and common descent via branching and test that against the dependency model, then the dependency model wins. Taking all of the other problems away, all the other issues we've talked about, that's not how evolution works. There are other mechanisms, and you have to test your alternative hypothesis against the totality of evolutionary mechanisms rather than a small subset of those mechanisms. That's a problem. So to summarize the 2018 paper, Winston Ewert claims to show the dependency graph model is superior to common ancestry in terms of explaining the observed pattern of diversity. This fails because it's ad hoc and arbitrary in terms of how you employ the modules. It ignores many evolutionary mechanisms, specifically those that would generate a non-hierarchical pattern. He then assumes erroneously that such a pattern is a problem for common ancestry. It's based on incomplete genome annotations, which is going to invalidate the results. And it doesn't even utilize the same data set. It's not even comparing the actual sequences using the new technique. You have to use the new technique, your dependency model, with the actual data we use to do phylogenetics. And Winston Ewert doesn't do that. He acknowledges he doesn't do that, which makes me wonder why we're doing this in the first place, because if you're not even using the same data, the comparison is meaningless. But we're not done yet, because we still have to talk about the 2023 paper, which is called Aminograph Analysis of the Auditory Protein Prestin from Bats and Whales Reveals a Dependency Graph Signal that is Missed by the Standard Convergence Model. Okay, so there's one glaring problem here, and it's that he's still not evaluating genome sequences. Now he's using amino acid sequences rather than nucleotide sequences. This is better, but it's still not good enough, and we're going to look at why, aside from the problem of it's the wrong data set, it's another apples and oranges comparison, right? Aside from that, there are two big reasons why amino acid sequences are not appropriate for the comparisons he's doing, okay? The first is that you could have very different amino acid sequences and get the same function. This is a well-documented phenomenon. Just one example is this 2023 paper, Sequence Structure Function Relationships in the Microbial Protein Universe. And this is figure 3 from that paper. Don't worry about reading all of this. I just want to show you that panels C, D, and E show examples of different structural motif, same function. If you read the legend down here, it says cases where the same function is generated by different structural motifs in different proteins, even though the proteins have the same fold. So you're getting different amino acid sequences, but you're getting the same function. So using amino acid sequences is inappropriate because in things that share common ancestry, you can actually get quite different proteins where you preserve the function, but you can have mutations in the sequences that change the amino acids in such ways you preserve the overall function of the protein, right? So in the common ancestry model, we can look at that and say, oh, that makes sense. In the dependency graph model, you would put a module in there to explain those differences, but we don't need to do that. Common ancestry does that just fine based on what we know about the constraints of protein structure and function. And then kind of the other side of the same problem is that because of the redundancy in the genetic code, you can get many different DNA sequences resulting in the same protein, right? So there are 20 amino acids that are used in Earth biology to make proteins. All but 2 of them have more than one codon, codon being a series of 3 nucleotides in DNA and RNA that code for a specific amino acid. So you can have 2 proteins that are very similar in terms of their amino acid sequence with vastly different DNA sequences, resulting in those structurally and functionally very similar proteins. Again, this is going to look like a high level of divergence when you're looking at it and at the amino acid level or at the nucleotide level, but this is perfectly explainable via common descent due to the redundancy in the genetic code, whereas Ewert would attribute that to a module, right? Saying, oh, that explains these different things in these different lineages, whereas actually common ancestry provides a perfectly fine explanation for that without the added complexity of a designer and the resulting modules, right? So the redundancy in the genetic code also causes a problem for this analysis. Again, like, my response to this 2023 paper was, seriously, what's the point of this? Just use the actual genome sequences. In between the two, there were conversations with, like, actual evolutionary biologists with Dr. Ewert, like on the Peaceful Science Forum, for example, where people brought these concerns to his attention. And then years later, he releases this second article that doesn't address those concerns. Just use the genome sequences. Just do the real comparison that everyone does when we do phylogenetics. I don't know why that hasn't been done, right? The first paper was 2018, then it was 2023, now it's 2025. Just do the real analysis and compare apples to apples and show that your model is better. Honestly, just do it already. All right, so let's summarize the 2023 paper. We're still not doing the relevant comparison, and, like, honestly, nothing else matters. If Ewert isn't testing the dependency graph model with actual nucleotide sequence alignments, it has zero relevance for the question of which model better explains the extant pattern of diversity. Just test it with the real data. that we use to do phylogenetics. That's all I'm asking, please. So we can wrap this up here. I know this was a long one. Thank you all for sticking it out this far. Dr. Winston Ewert claims his dependency graph model better explains extant patterns of biodiversity than common ancestry. To date, he has not actually evaluated this model with the relevant data set. And when I say to date, I mean the end of 2025. He has not done this analysis or this comparison with nucleotide sequence alignments, which is going to be the bare minimum that he needs to do. None of the other problems, of which there are a lot, actually matter, but all of those other problems are also problems, right? Assuming any violation of the nested pattern is a problem for common ancestry, ignoring the mechanisms that lead to that, relying on incomplete genome annotations, all of that stuff, those are actual problems that need to be fixed. But the big one is that he needs to test his model using the right data set. Use nucleotide sequence alignments. Until he does that and addresses that other stuff, nobody has any reason to take this idea seriously as an alternative to common ancestry as an explanation for extant biodiversity. Whew, we made it, everybody. Thank you all for watching. So dependency graphs, what did we learn today? Dependency graphs as a better explanation for biodiversity than common ancestry, that is a creation myth. Thank you all for watching, everybody. Please remember to hit the like button, leave a comment, subscribe. If you become a channel member, you get stuff like this pre-recorded right away. You don't have to wait for the public release. Thank you all again for sticking it out this far. I appreciate it. Don't get fooled.